Good afternoon, everyone, and welcome to today's session. So, we're gonna start our revision session in like 5 minutes so that people can join in. Okay. as I mentioned earlier, we're gonna start our remaining part of the revision today, and then we might, if the time permits, we might also discuss a new topic that will be action potential. Okay, so we can start our revision. So in the last class we revised our basics of amino acids, then proteins, then peptides, right? But at the end of the day, the course which we are undergoing belongs to membrane, so everything has to be studied with respect to the membrane. So we know what amino acids are, what proteins are. There are certain basic biochemical properties which can be exploited if we have the sequence of a protein, right? Based on that, for example, I'm talking about hydropathy index, right? Hydropathy plot, hydropathy profile, and all that. Based on that, if we have a sequence, we can actually take a guess that what part of this protein will be in the membrane. So one question which I want you guys to think upon is, can we do this similar analysis on cytosolic proteins, right? So cytosolic proteins won't be in the membrane, right? They will be in the cytosol itself, inside the cell, correct? So hydropathy profile we have discussed only with respect to the membrane protein. And what hydropathy profile is, or hydropathy index, it basically tells if there's a stretch of hydrophobic amino acids around maybe 20, 25 or 24, less than 20, 24 amino acids together, the chances are that this protein might be a transmembrane protein, okay, or it will be in the membrane itself. Can we speculate such behavior if I give you two protein sequences, right? One will be of a transmembrane protein, another will be from a cytosolic protein. So can we distinguish, just looking at the sequence of a protein, whether this protein will be a transmembrane protein or the other protein may or may not be a transmembrane protein? Okay, to answer this question, I can give you a hint that always remember what property we are exploiting to understand the protein behavior by looking at the sequence. Okay, it is that there will be a stretch of hydrophobic amino acids. And why we need a stretch of hydrophobic amino acids? Because that stretch of amino acids need to traverse the membrane, right? And membrane is lipidic in nature, hence hydrophobic. So only hydrophobic residues will be energetically favorable inside the membrane. Okay, so by the end of the class we can discuss this, or I guess it's a pretty easy question and you guys can easily give answer to this question. So we'll start our revision class for today. So as we have discussed about the protein, now we'll discuss about the protein which is specific to the membrane, right? So this is the representative diagram of a transmembrane protein or a membrane protein in general, say. So if we look such representation, we can say that this will be a transmembrane protein. If we are looking at a representation of a cartoon representation of a protein inside a membrane, we can say that it will be a peripheral protein. Okay, so there are 4 different types of proteins which are present in the membrane. They are peripheral, amphitrophic, integral, and lipid linked. These are the major classes. Now they have further subdivided these into multiple subclasses. So peripheral proteins, they can be bound to the protein or they can bound to the lipid. We will see in the coming slides. Okay, then amphitrophic, then integral. Integral further has 2 classes: endo-integral protein, ecto-integral protein, and transmembrane proteins. Okay, then last but not the least, we have lipid linked proteins. So lipid linked proteins can be classified further depending upon what kind of linkage is associated with the protein. It can be myristoylated, palmitoylated, prenylated, GPI anchored or linked proteins. Okay, so we'll see in detail in the coming slides. Yeah, so a basic revision as usual so that we can heat up our engines for better understanding. Yeah, here it is. I have already started this video so that we can save a little bit of our time. Let me share my screen with you guys. Yeah.
In this video, we're going to explore membrane proteins. Did you know that the cell membrane can be composed up to 75% protein? So most cell membranes have about 50% or less protein. And proteins are there because the cell membrane uses proteins for pretty much everything that it does, all of these cell membrane processes that it performs. So just to remind us what a cell membrane actually is, a cell membrane is made up of little things that look like this, which are called phospholipids. And they come together and form what we call a lipid bilayer. So over here, I've pre-drawn a lipid bilayer, and it'll look something like this. It'll be made up of a lot of these small phospholipids that we've drawn above, and it'll make up our bilayer. So you can see that there are 2 layers of these phospholipids. Now, there's 2 major types of proteins in the cell membrane. The first can look something like this. And this can appear anywhere in the cell membrane, and there are usually quite a few of these throughout the entire cell. So this is what we call an integral protein. You'll notice that it's called an integral protein because you can think of it like it's integrated throughout the entire membrane. Another type of protein that we might encounter might appear on top of the membrane. Occasionally, it might be slightly into the membrane, and it can also rest on top of integral proteins. And this we call peripheral proteins. And the reason why we call it a peripheral protein is because it's on the peripheral or the outside of the cell membrane. The difference between peripheral and integral proteins is that integral proteins are really stuck inside the cell membrane. As you can see in this picture, the integral protein is really inside the membrane, and as a result, it'll be very difficult to remove. Peripheral proteins kind of attach and remove themselves from the cell membrane or from other proteins. They generally are there for different cell processes. So for example, a hormone might be a peripheral protein, and it might attach to the cell, make the cell do something, and then leave. Peripheral proteins can also exist inside the cell on the cell membrane. Another type of protein is extremely rare, and it can appear inside the cell membrane like that. And we call this a lipid-bound protein. Why might you think a lipid-bound protein is so difficult to find, so rare? Well, the reason why is because proteins are there to interact with the outside environment. And lipid-bound proteins are stuck on the interior of the cell membrane itself. So it can't really interact with the outside of the cell or the inside of the cell. So it doesn't really serve a big function in terms of the cell membrane performing its duties. We're going to spend a little bit of time talking about 2 types of integral proteins that are extremely important because these 2 proteins are found all over the cell, and they help the cell maintain homeostasis or balance. The first type can look something like this. Again, this is an integral protein. What do you think this protein might be used for? This isn't 2 proteins, it's actually 1 protein with a hole through it. Well, this protein is actually used to allow things to pass through the cell. We call this a channel protein. And like the name kind of implies, there's a channel or hole inside the protein that lets things pass through. So for example, if there is some sort of ion, let's say this is an Na plus ion, a sodium ion, this is outside the cell. And the cell at this point really needs these sodium ions to perform a really important process. So what the channel proteins do is they'll allow these outside extracellular ions into the cell. And normally, these sodium ions wouldn't be able to pass through the cell membrane just by themselves. These channel proteins allow our bodies to take in different materials from the outside environment into our cells. What they can also do is they can also do the reverse. So let's say your cell has way too much sodium and it needs to get rid of it. So channel proteins can start pumping these out. Channel proteins generally don't require energy, so there's no energy needed. Sometimes we call energy ATP. And another thing that's special about channel proteins is you'll notice that it'll go with the concentration gradient. So out here there's a lot, and inside there's very little. So it'll pump from where there's a lot of sodium into where there's very little. So it'll go what we call down a concentration gradient. Concentration gradient. The second type of very important integral protein is called a carrier protein. And like the name implies, it carries substances into the cell. I kind of picture it like a baseball glove, like this. So if there's a molecule that's outside the cell, and the cell actually needs this molecule, so what the carrier protein will do is it'll actually protect this substance so that it can enter the cell safely. It can also do this in reverse. It can take something inside the cell and pump it outside the cell. And this type of protein is really important because unlike
right, so I hope the concepts are brushed up now. Let me get my laser pointer. So we'll discuss these type of proteins one by one. So as the first one indicated, peripheral protein, as the name suggests that they will be associated in the periphery of the membrane. Now as membrane is a bilayer, these peripheral proteins can be associated on any side, right? Our peripheral proteins are held to the membrane surface primarily through electrostatic or hydrogen bonds. They can be divided into two basic types, those that are attached through charges on the surface of integral proteins and those that are attached to the anionic head group of a phospholipid. Peripheral proteins can be removed easily and cleanly from the surface of membrane by either altering the pH or the media salt concentration. By definition, a true peripheral protein exhibits little or no interaction with the membrane hydrocarbon interior and is removed from the membrane without attached lipids. Classic example of peripheral protein includes cytochrome C, myelin. So from this, another question arises: can we distinguish a peripheral protein from a cytosolic protein if the sequence is given to us? Okay, think about it. Next type of membrane protein is amphitropic proteins. So amphitropic proteins are a relatively new class whose importance is rapidly becoming more apparent. Unfortunately, there is a lot of overlap between the peripheral proteins that are bound to membrane surface phospholipids, integral, ecto and endoproteins and amphitropic proteins. We have not discussed these classes yet as it is a revision, so you might remember where we are heading with this discussion. Okay. Amphitropic proteins can alternate between conformations that are either water soluble or weakly bound to the membrane surface through a combination of electrostatic and hydrophobic forces. Since the membrane interaction is reversible, an amphitropic protein coexists as both a free globular protein and a membrane bound protein. The water soluble form is converted into the membrane bound form following a conformation change induced by phosphorylation, acylation or ligand binding that exposes a previously inaccessible membrane binding site on the protein. Membrane interaction is often based on production of a hydrophobic loop of amino acids, sometimes an alpha helix that can penetrate into the lipid bilayer. So this is a very important point. This will be our basic theme whenever we're gonna say that a protein is functioning. Earlier it was not functioning, but later it is functioning. So why I am saying this? If you look at all the mechanisms which are known till now, how proteins work, right? If a ligand is coming, then the protein is conducting it. If another ligand is coming, the protein is not conducting it. Something changed somewhere and the protein starts functioning. Earlier it was not functioning, yet it was present in the system, but it was not working at the end of the day, right? So if we try to boil down to the basic concept of protein functionality, it will always be associated with a structural change, right? Irrespective of whether it's a membrane protein or a non-membrane protein, stimulus will always be associated with changing the structure of the protein, and that conformation change or the structural change will leads to the further downstream things which that particular protein is supposed to do. Okay, so these changes, how these changes can be made, we have read about amino acids, proteins, right? So we classified amino acids based upon their side chain. There were 5 major classes. So imagine if somehow we change those properties. If it's an acidic amino acid and we put that acidic amino acid in a basic environment, or say if it's a non-polar amino acid, we put it in a polar environment. It can be any condition, right? So based upon the condition, proteins / amino acids will change their biochemical properties, right? These biochemical properties will alter the structure of the protein, and hence the desired function can take place. It's a bit difficult to understand because I'm simplifying a lot, right? There's a lot of knowledge present in the arena of proteins, but in actuality, according to my understanding, this is the diluted version of how proteins function. Okay, next type is integral proteins. It is estimated that 20 to 30% of cellular proteins are integral, and these proteins are tightly and permanently bound to the membrane, while peripheral and amphitropic proteins are weakly bound to the surface of membranes and can be readily dissociated, producing lipid-free globular proteins. Integral proteins are integrated into the membrane hydrophobic interior and can only be removed by more drastic means. Historically, integral proteins have been separated from membranes through acetone and other non-polar organic solvent extraction methods. Although thousands of globular proteins have been crystallized and their 3D structures are actively available in a database, we have very less transmembrane protein structures present in the database. Okay, because of this difficulty, because we cannot extract the protein intact because it is so well preserved in the membrane that whenever we try to, with the technologies which we have, when we try to take out the protein from its natural environment, it comes out with the lipid membrane itself, right? The lipids through which it is surrounded. This is one major reason why membrane protein isolation and purification is very difficult. Endo and ectoproteins. Endo and ectoproteins do not traverse the entire membrane. Instead, they are inserted into the membrane's hydrophobic interior from either the cytoplasmic side or the extracellular side. An example of such protein is cytochrome B5, and we have seen the representative videos and certain examples also, right? This is an important class of protein. All classes of protein is important, but when we talked about the functionality of a membrane, so most of the proteins for which we have studied the functionality, it belongs to the transmembrane class of membrane proteins. A transmembrane protein must span the entire membrane with segments exposed to both on both the outside and the inside aqueous space. So the representative structure which we were talking about, which we saw at the beginning of our today's discussion, it was for the membrane protein, right? The membrane that must be spanned is composed of a lipid bilayer that can be divided into 3 sections. The inner hydrocarbon region, approximately 27 to 32 angstrom thick. A very narrow boundary region between the hydrophobic inner core and the hydrophilic interfacial region is approximately 3 angstrom. You will understand when we see the representative figure, because we know that the lipid bilayer thickness is to some extent, like it's 4 to 4.2 nanometer. Hence, if a protein is transversing this, the protein must span the whole membrane, right? So this is a generic idea of the size of a transmembrane protein based upon the known fact that membranes are this much in thickness. All membrane proteins are 100% asymmetrically distributed with respect to the membrane and do not flip-flop across the membrane. Therefore, transmembrane proteins are locked in place. A characteristic of many transmembrane protein is the presence of tyrosine and tryptophan at the aqueous interface. You remember we discussed this point when we were discussing about the aromatic amino acids. And there were aromatic amino acids. If you remember, there was a tyrosine residue, right? So tyrosine, though being hydrophobic, so it will prefer to stay in the hydrophobic environment, but it had one OH group which would like to make hydrogen bonds. Hence, I mentioned that these type of amino acids will tend to stay at the interface between the lipid bilayer and the interior of a lipid membrane, right? These amino acids serve as interfacial anchors that can interact simultaneously with the membrane hydrophobic interior and the aqueous exterior. Also, for reasons not entirely clear, the cationic amino acid lysine, histidine, arginine occur more commonly on the cytoplasmic side of the membrane. We somehow have idea about this point when we talk about the action potential part of it, but this property is only present in the excitable type of cells but not in all cells. Okay, so these are the different types of transmembrane proteins at the end of the day, and this was the hydropathy profile of bacteriorhodopsin, and you can see the sequences also which very beautifully coincide with the hydropathy profile. Correct. Then we discussed about the lipid anchored proteins. As I mentioned, there will be a lipid associated with the protein which will embed itself into the hydrophobic environment, and based upon the type of lipid anchors which we are discussing, we can have different lipid anchored proteins. Then we discussed about different functions of membrane proteins. As I mentioned, the functions which we have discussed majorly belongs to transmembrane type of proteins, right? So 6 major functions are enzymatic activity, signal transduction, transportation, cell-cell recognition, intercellular joining, and anchorage or attachment. So let me share my screen one more time, because we are not discussing all the functions which are associated with membrane proteins, but we have discussed major few of the major functions which are performed by the membrane proteins. Okay,
this is an excellent title slide showing the 6 major functions of membrane proteins. I suggest you watch the video and you come back to this and use this kind of as a self-quiz, and can you understand all these images? Right now they may not make a lot of sense, but if you look through the slides, I have one on each, and then come back to this and hopefully it'll make more sense. Starting with the first one, transport. So one of the major functions is transport. This slide may look familiar from one of the previous ones. It is an important function. There's many different forms of transport. In general, here on the left side of the image, we have our proteins that span the membrane. These may provide hydrophilic channels across membranes that is selective for a particular solute. In this case, it's allowing the little green molecules to come through. On the right side of the image, these carrier proteins require energy. They're actively grabbing these more oval molecules and transporting them through the phospholipid bilayer. Another major function is enzymatic activity. The protein built into the membrane may be an enzyme with the active site exposed to the substances in adjacent solution. In some cases, several enzymes in the membrane are organized as a team that carries out sequential steps of metabolic pathways. So we see the example here. We have lactose, and remember ACE at the end of any word indicates it could be an enzyme. In this case it is. It's taking our lactose and breaking them apart, this disaccharide from carbohydrates, into its 2 monosaccharides, a glucose and a galactose. So here's our disaccharide, lactose, being broken apart into its 2 monosaccharides, glucose and galactose. And this is because of enzymatic activity that's embedded into the membranes. And remember, enzymes are specific types of proteins. All enzymes are proteins, but not all proteins are enzymes. Signal transduction. So a membrane protein may have a binding site with a specific shape that fits the shape of a chemical messenger, such as a hormone. The external messenger or signal may cause a conformational change in the protein receptor that relays that message inside the cell. What does that mean? That simply means we have a receptor protein embedded in the cell membrane here. It's binding to a specific hormone, and that is basically leading to secondary messages and a response in the cell. Remember, our active site is very particular on what it's binding to. It's a very specific shape. Gave the example in a lock and key model. Another major function, there are cell-to-cell recognition. Some glycoproteins serve as identification tags that are specific to recognize by other cells. This is very important in the immune system. We want to know what cells are self and what cells are foreign. So these glycoproteins are very important. It's just highlighting one right here for cell recognition. So cells can recognize other cells for binding purposes, could be for attacking purposes, to recognize what self versus something that's not self might be. The fifth one along those same lines are intercellular joining. Membrane proteins of adjacent cells may hook together in various kinds of junctions, such as gap or tight junctions. This can help allow cells to closer communicate, can allow for easier exchange of materials. So this intercellular joining is another major function of our membrane proteins. 6, attachment of the cytoskeleton and extracellular matrix, which is abbreviated ECM on some occasions. These microfilaments and other elements of the cytoskeleton may be bonded to membrane proteins. So we have our microfilaments here of the cytoskeleton. Here's our proteins kind of binding and holding everything together here. This function helps maintain cell shape, stabilize the location of certain membrane proteins. Sometimes we want our proteins to be able to move through the fluid mosaic. Other times we want them fixed in place. And this can help maintain them in specific locations. Proteins adhere to the ECM and can coordinate extracellular and intracellular changes. Remember, ECM is referring to extracellular matrix. This kind of shows just a graphical representation of that. That is the 6 major functions, and remember to go back to that beginning.
Right, so that was the brief of it, but we personally have discussed 2 major functions, which is transportation and signaling, right? So we're gonna come back to that also, but at the end of the day, the basic principle which is guiding that why we actually require proteins in the membrane. When membrane is self-sufficient, then why do we actually need proteins? Because making proteins will be energetically too much cumbersome for a cell, right? You have to do transcription, translation, and then assemble these proteins to the membrane. So it must be very important for a cell, then only it is doing this function. So what is the actual need of having proteins in the membrane can be easily understood from this figure. So what I mean by this, as you know that membrane is made up of lipids, right? It's a bilayer. Lipids make hydrophobic environment. In chemistry 101, we know that like dissolves like, correct? And basic diffusion is a slow rate, and the time scale on which we work, for example, that as soon as the stimulus comes, we react to it. Excuse me. Just by simple diffusion, these changes might not happen in the required time scale. Hence, we need certain passages in the membrane so that it can be enhanced. The time of transportation, the time of signaling, the time of enzymatic reactions or biochemical reactions can be lowered down so that we can see effect in a lesser time, correct? So in this figure, what it says that if a molecule is of gaseous type, such as CO2, N2 or O2, it can be easily permeate via bilayer. Hence, they do not actually require any assistance of proteins for this purpose. And the rate through which gases get transported, passively transported, it's enough for the cell to sustain. Then we have another category of small uncharged polar molecules, right? Though molecules are small, hence it will be easy for these molecules to pass through the membrane. But they are also polar in nature, right? So this property will definitely pose something or some resistance in transportation, right? So if a molecule is small and polar, chances are that it might permeate and also it might not permeate. Then we have large uncharged polar molecules. Definitely this class of molecules won't be able to cross the bilayer on their own, correct? Then ions, again because they have polarity associated with them. Then charged polar molecules, like bigger molecules and other molecules which have polarity. So for them to transverse from one side to another side, we need transporters. So transporters are a type of membrane proteins which help in transportation of substrate. And what kind of substrate? Substrates which are polar in nature, which are larger in size. So majorly these type of molecules are transported via membrane transporters. So there are two basic types of transportation which is present in the living system. It is active transport and passive transport. So in passive transport, molecules will transport from their higher concentration to their lower concentration. Now two conditions can be fit in this. If the concentration is high, the molecule can transport on its own without the assistance. So what kind of molecules will they be? They will be of gaseous types, right? Non-polar small molecule. And that is called simple diffusion, correct? But on the other hand, if the molecule is, imagine, small, but it is also polar, hence it needs assistance in transporting, in like getting from one place to another place across the membrane. Hence they require proteins. So wherever there is transportation of molecules along their concentration gradient and protein is involved in it, that type of transportation is called facilitated diffusion, right? Now if we talk about active transport, in active transport, active, as the name suggests, that there will be association of energy with it, right? So active transportation, wherever there is usage of energy, and energy will be used only when we need to transport molecules against their concentration gradient, right? Hence if a protein is present and it uses direct ATP or energy for transporting molecules from its lower concentration to higher concentration, it is called primary active transport, correct? On the other hand, another type of active transportation can be understood. For example, we need to transport a molecule which is in lower concentration, right? So definitely we need to have an active transportation because we are transporting from lower concentration region to higher concentration region. So we need energy. But what if this energy is not coming from the ATP molecule or the energy molecule, but is actually coming from another substrate which is already present in higher concentration in the same site and can be easily transported on its own with the help of protein along its concentration gradient. So it is actually using secondary energy from this particular substrate, hence it is called secondary active transportation. Okay, these are the basic properties which are associated with these different type of transportation. For example, requires specific protein. So only simple diffusion will be a type of transportation in which we do not require any specific protein. Solute transported against its gradient, this property will only be associated with active transportation, and active transportation is of two types: primary active transporter and secondary active transport. Coupled to ATP hydrolysis, this is a characteristic of primary active transport. Okay, then driven by movement of a co-transported ion down its gradient, it is a property of co-transport or secondary active transport, and these are the specific examples of it. As the transportation is of different types, hence the transporters are of different types. If we talk about channels, these are just like a opening in the membrane, and they open and close based upon the stimulus, and they mostly transport substrate from its higher concentration to its lower concentration. Second, we have transporters. Transporters are the one which actually transport substrate by involving a conformational change in the protein. It can be of three types: unipolar, sympolar and antipolar. Unipolar, as it is represented here, will transport one substrate from its higher concentration to its lower concentration. Sympolar, sim means together, indicating that it will transport two substrates, one along the concentration gradient, another against the concentration gradient, but both transportation of a molecule will be on the same side. Then we have antipolar, as the name suggests. Again, it will be from lower concentration to higher concentration, but the transportation side of these two separate substrates will be opposite or will be anti. Then we have ATP powered pumps. This is an example of primary active transport. It will use ATP energy or energy molecule to transport substrate from lower concentration to higher concentration. Okay, so in the next class we gonna discuss again about the sodium potassium channel and action potential. Okay, feel free to drop me mail if you have certain queries. As I've already mentioned, I've started uploading the videos on the YouTube. By the next class I might share the link, or I actually figured out how to upload on the Moodle platform, so you guys can access from there also. Okay, thank you.